start recording now. And this is the last day of the spring school. It's April 10th, 2021. And the first speaker is Mirjerta Pasha from Arizona State University. She will talk about an optimal experimental design approach to learn parameters for solving large-scale inverse problems. So Mirjerta, it's your stage.
Hello, everyone. I would like to start by thanking the organizers for this spring school and also for the opportunity to give a talk. Today, I'm going to talk about some work that is focused on learning parameters for solving large-scale inverse problems. And this work is in collaboration. So part of this work is in collaboration with my PhD advisors, Alessandro Boccini and Professor Lothar Reichel at Kent State University. And the work, the current work on learning is with Professor Julian Chang at Virginia Tech, Matthias Chang at Virginia Tech, and Professor Silvia Gazzola at the University of Bath in the United Kingdom. And this work is done under the SAMC meetings group work. So I'd like to start with a motivation first. So the first part of my talk will be on inverse problems. What are inverse problems, and what are possible ways that we can solve inverse problems? And then the second part will be focused on the learning approach that we are currently working on. So there are many applications actually of inverse problems, and one of them is computerized tomography. So we can think about it that we have a non-transparent object, and we are interested to look inside those objects. For example, we want to look inside the brain and so on. And we can use techniques like computerized tomography. What happens, we shine some X-rays, and then with mathematical functions, there will be produced a sinogram. And then using this sinogram, like the information, which is basically a matrix where we have pixels, 0 for black and 255 for white, of course, depending on the scale. Here comes our work where we want to develop algorithms that can reconstruct the images or the interior of the object fast and also with high accuracy, because better is accuracy, then easier is for doctors and so on to look for anomalies. So another application I would say is image deblurring. So suppose that we have an image that has blur and noise that may come from different applications. Then we're interested in finding an approximate solution. So to go to the mathematical setup of this problem, then we can model this type of problems in linear or nonlinear. In this talk, I'm going to be focused on the linear problems, but of course you can extend this to nonlinear. So we have an operator A over here that models the forward problem. X are the desired parameters or the solution that we are looking for. B are the observed data or the measurements, and B true are the data without the noise. And actually in the observations that we get or the measurements, we cannot get them without noise. And in general, there are different types of noise that contaminate our data. So might be Gaussian, might be impulse noise, or a mix of them. And the goal is that given those data and some information about the forward problem, then we want to compute an approximate solution. Well, this type of problems are ill-posed because the solution may not exist, may not be unique, or it does not depend continuously on the data. And we see over here that what happens is that if we consider finding the naive solution over here, then what we get back is something that is far away from even the blurred image, not even close to the true image at all. So what do we want to do? And what happens actually over here is that these problems have stability issues, meaning that small changes that we are going to make in our data will cause very big changes in the solution. So to counteract the effect of noise and those difficulties coming from these problems, we consider regularization methods. And one type of the regularization method actually is based on this type of functional, where this part over here is called the regularization term. And this operator is the regularization regularization parameter, and the first part over here is called the data fidelity term that take care about the noise. So over here, we choose different values of P and Q to accommodate different choices for the noise, and also different choices related to the characteristics that we want. So for example, if we have solutions that we want to preserve edges, or we have solutions with let's say piecewise constant derivative and so on, then we want to make sure that we can provide this kind of solutions where we solve the problem. Choosing P and Q equal to 2 goes back to classical Tikhonov and so on. But we are mostly interested over here for the cases where P and Q are less than 2, and ideally, so for example, I'll give a motivation of this. So for example, if we want to reconstruct solutions that are sparse, then ideally we would like to reconstruct the L0 quasi-norm over here that is represented by this black line, but this is NP hard, so we want to find, one can use the best convex approximation over here by choosing Q equal to 1, but as we see from this graph, better if we choose those lines closer to 0, then the approximation is going to be better. But of course, there are introduced other difficulties in the problem. Here is an illustration where we show that if you have in your data, if you have, of course, some blurred images and some type of noise, and especially over here, I have a mix of impulse noise and the Gaussian noise, then if you use the standard P, by solving by standard Tikhonov, then you're going to have issues when you have the reconstruction. While if you choose different values of P, and ideally, if you learn those values of P and Q over here, then you will get a very nice reconstruction. So basically, the type of methods that I've been working on are based on the generalized Krylov subspace and the majorization minimization type of method. So the whole idea is that if you have a functional that is not differentiable, then you can consider a smooth version of this functional. And by considering the smooth version of this functional, then you can consider a sequence of these iterate that converge to a stationary point, and then at each iteration, you can find the unique minimizer of the quadratic tangent majorand. This can be done by defining some weights, and then once you have the weights, you can transform your problem to some basically to some L2 problem that can be solved. This can be computationally very expensive. So what we do to remedy this type of difficulties, we solve this in a generalized Krylov subspace. So we start with a small subspace, and then at each iteration we enlarge the subspace until the convergence. Now let's go back to the goal over here. So this was kind of the background or the type of methods that we use to solve this approach, this type of problems. When we have the training data available, then we want to learn the parameters to solve this type of inverse problems. So learning actually has been used in different approaches, for example, to learn regularization parameters, to learn optimal filters, to learn parameters. I've cited some references over here, but there are many others out there. So given a set of observations and also a set of noise samples, then if we select only one of these here, just to show that we have to solve an inverse problem, then the problem that we have to solve is this problem over here. So given B and A, then we have to determine that solution. So in this learning approach, what we consider variation approaches that can be written in this form over here, and I want to show that. So what we use is a bi-level optimization actually, where we try to minimize the Bayesian risk, the base risk, subject to finding the solution that solves this minimization problem over here. And the design parameters over here are those theta, and it's the expected value, and we model the true solution and the noise as the random variables. So to summarize all the idea, the design problem can be thought as a bi-level optimization schema. And the goal is to compute those parameters from the outer problem where this X is obtained by solving the problem 7 over here. So there are different approaches actually. So when the distribution for the X2 is unknown or is not obtainable, then what we consider the empirical based risk design problem. So for example, given a set of training data, given a set of noise samples, then we simulate a data set and then we obtain those reconstructions and then we define the empirical based risk optimal experimental design problem to be defined by this 9 over here. So of course, there are challenges with the bi-level optimization framework, but to address and to overcome those challenges, we consider constrained global optimization for the outer problem. And then in the problem that we are trying to solve, there are other difficulties that come from for different choices of the noise and also the characteristics of the problem. So for example, for sparsity and so on, the problem might become more difficult to solve. So to accelerate the convergence, what we use the information from the training data to define different transformations. So for example, we use the sample mean, or we use the weights that we can obtain by our training data. And in this approach, actually, we consider this a very generalized approach because we can learn regularization parameters, we can learn regularization matrices, we can learn different type of kernels, and we are considering maternal kernels, square exponential kernels. We can learn the fractional Laplacian actually is a generalized, it's a special case of this general framework that we are considering over here. And to be considerate for time, I will just show over here that just a simple example. So consider that we have 30 training images over here with these noise samples coming from the levels 1% to 5%. What we do, we learn the regularization parameters in the regularization matrix. And then we, in the 10 validation images, we validate the result. And what we obtain back is images on the validation set with average relative reconstruction error, like around 10%. So to summarize, this is like the approach that we are working on is a general framework, and the focus over here, or the novelty, is in learning errors that might come from the measurement, but also from the errors in modeling the forward problem. And with this, I would like to thank you again for the attention and for the space. And this is all for my talk. I'm ready to take any questions.
Okay, thank you very much. Any questions from the audience? Or, I have a question. The thing is that it is kind of very well known settings, but the thing is about the matrix A, when you consider it, it also could be subject to noise.
Yeah, sure. Like the operator might also have noise, and we consider that case in our framework. And what we found actually interesting over here, well, this also depends on the noise in your data, because what might happen is that, so what we try to do here by choosing different values of P in our functional, let me go back to my functional over here. So by choosing, suppose that you don't know the true A, but you have errors in your operator, and then by choosing different values of P over here, then you can take into account those errors that come from in the modeling operator over here, A.
Okay, thank you. Other questions?
Can I ask you something? At some point in your algorithm, you needed to expand the spaces.
Yeah. Mm-hmm.
How do you do that? I mean, there is a universe of ways of expanding the spaces. Do you have any kind of systematic way that actually tells you you will benefit from a specific expansion in a quantifiable way?
Yes, yeah, that is a very good question. And sorry that I didn't go into details on that. So there are different approaches actually in the way that you can extend the subspace. So, and also depends on the type of curl of subspace methods that you are using. So for example, if you use the generalized curl of subspace method that I'm using over here, when you compute a solution of this problem over here, so let's say at some iteration k plus 1, you have this solution xk. So what we do, we compute the residual at that iteration. So so far we have a subspace. So we start with a fixed subspace, but of course you can start by 1, and you can define the residual. And we use the residual vector. We orthonormalize that, and then we add it at the solution subspace. And in that way, we make sure that every vector that we add will form a nice subspace for our solution.
This is okay. Maybe that sounds natural, but on the other hand, the residual lives in another space than the solution. Actually, it's subject to a mapping.
Well, actually, I have also tried to expand the solution subspace by the solution itself. So like normalizing the solution itself and then expanding it. But I haven't seen too much of a difference in the numerical computations, because we were looking for different ways to also speed up, because there are also issues when the problem becomes very large. There might be issues in the solution, like the methods that you are using and so on.
Okay, thank you very much. I have to stop the discussion here because we have the next talk. These are fast talks. So, okay, stop sharing.
Yeah, thank you.
Craig can start here. Okay, the next talk is by Craig Chen from, this is from Duke University, and he will talk about policy gradients for Lqrs with nonlinear controls.
Thank you for the introduction, and also thank you for hosting me for this talk. So disclaimer, I am a bit new to this. I'm actually still an undergrad at Duke. I'm in my 3rd year. So any hiccups, just a disclaimer. So this talk is about some recent work with my mentor at Duke, Andrea Agazzi. It's focusing on trying to prove convergence of policy gradient algorithms in the setting of the Lqr when we parameterize the function approximator for the policy with some nonlinear functions. The context of this work is in the past few years, a bunch of people have been using the Lqr as a proxy for more general reinforcement learning settings. Because the Lqr is very well studied from the optimal control theory perspective, so we know a lot about it, so we can have certain expectations going in when trying to prove behavior of reinforcement learning algorithms. The most directly related work to this is by Mirjerta at University of Washington. They published something in, I think, 2018, 2019 that proves convergence in this setting, but for linear controls. And this is actually a very important difference. So the motivation is, well, I mean, reinforcement learning has been super popular for just like general problem solving. I mean, DeepMind has been doing a lot of stuff with reinforcement learning, OpenAI, like, and it always seems to work. Granted, their problems and like their function approximators, so their like neural networks, are much more complicated than what we'll be studying in this talk, and the problems themselves are also like much more poorly behaved. But from a like, kind of statistical point of view, everything that they've done, like there's no theoretical guarantees at all that it should work. And so naturally we're asking like, well, can we show that this works even in the simplest setting, such as this Lqr? There's very few guarantees so far. So the Lqr, or linear quadratic regulator, is a standard problem from control theory where you have dynamics that are linear in the state. So this xt plus 1 is just a linear function of xt, and then ut, which is the action that you choose at time t. We're interested in the noiseless case here, but if you add zero mean noise, it doesn't really change any of the answers, so it's okay. And we're also interested in the infinite time horizon problem of minimizing this quadratic cost. So it's quadratic in the states and in the action. And gamma in this case is some discount factor. And this discount factor will play an important role in the actual kind of proof technique that we choose. So conveniently, and so this is the important part about what I said about linear policies earlier, is that the optimal solution for the LQR is known to be a linear function of the state. So when you try to solve this problem and you just initialize your approach saying like, okay, we're considering linear policies, you have a pretty big head start because you know that your policy class is already like the policy class of the optimal policy. And then the two equations here are just, so we can also solve for the optimal policy without using any learning techniques or anything, but you can solve for P gamma just using some dynamic programming approach. It's nonlinear in P, but like there's a bunch of literature out there that shows that you can do this. Sometimes it might be a little bit slow, it might be unstable sometimes, but it still is doable. And that's the caveat, is if you know the matrices A and B, which govern the system. So the next main component of this is the policy gradient algorithm. So this, to make it very brief, is you're trying to learn the best action from any state in your environment. And so we can try to learn some function to say like, oh, you input a state and then you output an action. You can also do it by just like keeping a table of all the actions, but this is gonna like not work for like chess, for example. Like good luck storing enough states, storing enough data for that. So instead we can use function approximation because it's much less memory intensive. And we just update our function parameters via gradient descent on the cost function. And the only reason this is actually a viable approach is because of this thing called policy gradient theorem, proved by Richard Sutton and some of his colleagues like 20 years ago, which showed that you don't actually need information about the state distribution to compute the gradient with respect to the cost function. And this is useful because naturally the way in which you choose your actions also affects the states or like the experiences that you have as a machine reinforcement learning agent. But since we don't need this information, we can actually compute these gradients. So we just update the parameters to standard gradient descent. Step size is, I mean, you can choose whatever, basically. There's a bunch of stuff out there on that. But the important part of this slide is the fact that we're working with this kind of new parameterization of a linear combination of nonlinear policies. And the important part of this parameterization, and probably one of the limiting aspects of it, is that we require it to be linear in the parameters. Well, just by design, like if you look at it, it is linear in the parameters. But this is a property that we have to exploit a bit to prove our results. And this extends the class. So this kind of subsumes the previous results by Mariam Fazal, where if we consider the Fk to be just like the matrix of the n by n matrix, whatever the dimensions correspond to the state in action, with a 1 in like the ij component, then what this ends up being is like you can just rewrite this as a matrix that we're trying to learn, which is what they did. But in our case, you can also add in nonlinear terms. So I see that I'm running out of time soon, but so the main trick behind our approach is that, so learning is hard because you have to consider very far ahead consequences of your current actions. And in an intuitive point of view, it's like you wouldn't teach a child to start thinking about college in like kindergarten, right? You tell them first, just like figure out your life, like don't die. So you tell them to focus on very short-term goals before extending it to longer-term goals. And we try to take the same approach for reinforcement learning, where we initialize with a very small discount factor and iteratively solve these like harder and harder problems. So we run policy gradients until we converge to the optimal policy for this smaller discount factor. And then we update the discount factor to a slightly larger one. And the main technique behind this proof is we first prove that like this local convergence. So if the discount factors are close together, we prove that the optimal policies are close together, and then you get exponential convergence from like gamma to gamma prime when they're very close together. And then you can just string this together. So you iterate it a bunch of times. And all of this is kind of in this continuous, like infinitely small step size setting, but that's a direction for some future work actually. The last part is there are some examples. So this is a policy that satisfies our assumptions and fits into our policy class, but if you train it with discount factor 1 using standard vanilla policy gradients, it actually gets stuck at a local optimum. So I just have an interesting counterexample that took way too long to come up with. So I think the main highlights of this talk are taking this new perspective on reinforcement learning and in general this control problem of iterating a discount factor to converge to the undiscounted optimal. So we've been able to extend the linear results for linear policies to a wider family, and we're kind of working on extending this to be able to say something about neural networks, but that's still very, like, the proofs are very sketchy. Like, we still have to work out some of the details. But thank you everyone for listening to the talk. I'm sorry that I went a little bit over time.
Yeah, thank you, Craig. No, you exactly on time. So any questions?
So can I? So
Sure.
your choice of making this linear combination in the end for the policies is because you would like to have a proof.
Yeah, it's the assumptions for our policy class basically came out of like, what do we need for this proof to be true? Yeah.
Okay. Did you try something else?
Yeah,
Like, you could have more involved, but still manageable parameterizations for that policy.
Yeah, that's something that Andrea and I are working on right now, because we
Okay.
a lot of this was kind of work done during like a summer research program with Duke, and the results we proved back then was actually much simpler. It was just considering like a linear policy, like some matrix case, like some linear policy plus some like nonlinear perturbation. But we realized that like those assumptions were not necessary, so we just said like, what's the most general thing we can prove? And this important criteria of being linear in the parameters, we're thinking maybe we can extend to neural networks in the lazy training regime, where you have this like super wide network, and I think with the neural tangent kernel setting, like maybe we can say something in that setting, but we still have to kind of work out some of the details.
Thank you.
Thank you for your question.
Yeah, other questions? Seems not. Thank you very much.
Okay.
Very nice talk and on time. Could you please? Yeah. So the next speaker is Guanqun. So can you start sharing? Yeah.
So you can see my slides now, right?
Yeah. So.
Okay.
Yeah. Can you enlarge it a little bit, or that's what you want to do that, because it's kind of showing only.
Okay, from my end, I already show the full screen.
Oh, okay. That's
fine.
I
have the same problem when I do that. I can never have people seeing the full screen. I don't know.
Okay, that's fine. I think that it could be. We're a little bit ahead of time, but I think that it's good to start now, because if there are more questions, it's better to have a little bit more time. So the next talk is by Guanqun from University of Auburn University. Okay, estimation of the mean functions of functional data via deep neural networks. Please.
Thank you for the nice introduction. Today is I'm really happy to share my recent project on functional data analysis. So first, I would like to thank my collaborator, my student Shuoyang Wang, and my collaborator Dr. Zuofeng Shang from New Jersey Institute of Technology. And I would also like to say that this early version of this paper has won Inner Student Paper Award in this year. I would firstly like to briefly introduce the functional data regression model and introduce the DNN structures. And then I will focus on the method, how to estimate the mean function in the functional data analysis by deep neural networks. And also I will present convergence rate. And finally, I will show the result for the motivating data set. my motivating is Alzheimer's disease.
Can you move the slides? Is that you're talking only on the first slide?
Yeah, let me back to this.
Yeah, okay.
Maybe this is better because I am not sure when I use the full screen, a little bit delay. So can you see the page 4, right now, the brain image?
Now we can see it, yeah.
Yeah, sorry for the confusion. My motivating example is Alzheimer's disease, the brain imaging data. As you may know, that Alzheimer's disease is an irreversible progressive brain disorders. And this disease gradually destroy people's memory and ability to complete simple tasks. So when people have the disease, for the healthy brain, you can see that this is healthy brain. And when people have the disease, it usually takes place in the hippocampus. And for the mild disease here, you can see that the hippocampus, the brain tissue shrink a little bit. And when the disease getting worse, the neuron dies, and brain tissues shrink significantly. So although right now there is an efficient cure for this disease, but the earlier medical doctor can help the patient to diagnose the disease, the better treatment can give to the patient, and slowly the progress of the disease. So the patient can have better life qualities and also their caregivers. So how could we have a better imaging data set for telling the patient and doctor that the stage of the patient? So right now, the data set the medical doctor usually have is called PET imaging from the machine that we can have is we have a lot of pieces of the imaging from the patient at the same locations. So the question is, what's the underlying true structure for the health people? And also what's the underlying true imaging structure for the patient at a different stage? So when we summarize the data set into a model, we can use a model called the functional regression model. So you can view the imaging, each people's imaging as a data set as a subject. And the data set, each pixel we have is Yij. So I represent the I's patient and J represent the J's pixel in that brain 2D imaging. And this Yj can be written as the signal, the main function F0, and plus some individual variations here, the eta, and also plus the measurement error terms here. So the F0 is from RD to R. So the X belongs to the D dimension. So the D for here can be 2 dimension, but later we can also have D3, the 3D imaging. And so the expectation of the Yj equals to the F0. The eta here is a process. So that's introducing the individual curve variation. And here we assume it's zero mean Gaussian process. And epsilon I is zero mean measurement errors. So our key question is how to estimate the F0, the main function here. So because that's the true signal for the whole populations. In statistics, the functional data analysis, there are a lot of different ways to estimate the main function. But considering here the fx here, the x is 2D or 3D dimension, the current method has some drawback in terms of dimensionality. So here we are trying to borrow some advantage of the deep neural networks. So let me briefly introduce the DNN's structure here. So we define the fx. Here x can belong to the RD, and it can be represented as this composition functions or DNNs. So there are a lot of layers. In each layer, that is weight matrix W times x and plus the bias vector. And so this is the first layer, and outside this is a nonlinear function. Here we use ReLU activation functions. So we are trying to use this DNN structure to estimate the F function. And so we know that we can use very large DNNs. That means very deep and very wide. So therefore, the parameters here, the W and also the L, can be contains extremely large number of parameters. So in order to consider the computational burden and also overfitting problems, we also add a constraint to put a constraint on the number of non-zero parameters for the weight matrix and also the bias vector. So here the S is to control the sparseness of these neural networks. So in the following, we are going to use this sparse networks to find the function in this space and to approximate the F function. So before I introduce the estimator, let me briefly mention the structured composition functions we are going to use for the two functions. So here we assume like we have a function vector gi, and it's from RDI to the DI plus 1. And for each component, Gij here is beta I holder functions. And although this function is beta I holder functions, but the intrinsic dimension is Ti. So usually the Ti is smaller than the Di. So we assume the true underlying function space, the G consists of a composition of many G functions, GQ to G0. So the smoothness of this F function depends on the beta I. So depend on the GI, that's the smoothness of beta I, and also times beta I plus 1 to beta Q. So that's how we define the smoothness for the F function and also the intrinsic dimension. So as we mentioned, we are going to find a function in this DNN sparse network space and to estimate the F function. So we denote the estimator as F hat. So it's the idea is very simple. In the input layer, we just put our observed X. That's the observation time point or location point. So for example, if we consider the 2D imaging, that will input xj1, xj2 for all the different j. And output layer, that will be the y bar dot j, that we take the average across the subject at j's location. So therefore, this is how we train these DNNs. And then by doing the optimization of these problems, and we can find F hat. So that's our estimators for the true underlying mean functions. We also developed the convergence rate in terms of the L2 empirical risk. And we also find that if we ignore the log term here, the convergence rate is quite similar to the optimal non-parametric convergence rate, regardless we have D-dimension data set. As we mentioned, in the current literature, the non-parametric estimation for the true mean function, if it's high dimensional data, it will have the cursor dimensionality problems. That means when the D is large, the convergence rate is decreasing dramatically. But here, by assuming the F true mean function have the composition structures, and also by using the DNN method, our estimator can almost approach to the optimal convergence rate of the non-parametric setting. So let me briefly show the real data as we mentioned at the very beginning. We obtained this data set from the Alzheimer's disease neuroimaging initiative, and we have totally 79 subjects from the Alzheimer's disease group. And we have each patient, we have 39 sliced 2D imaging. And so that means we have 39 subject here. 39 by 95 by 95. So this is 3D imaging. And then for the method, we are using this 3D scans, and also we recovered the imagings from like at the 20th, 40th, and 60th locations. So that's our recovered online true structures, the imagings for the Alzheimer's patient group. So due to the time concern, I didn't show the simulation result, but you can see that for our real data analysis result, we will have very nice simulation result. So I skip that. And also, if you're interested in our method, you are welcome to download our R package from the GitHub website, and also our paperwork is available on the archive. So that's all for my presentation, and thank you.
Yeah, thank you very much. So any questions?
Yeah, thank you for that very interesting talk. But let me ask you this. In a way, you say the enemy is the curse of dimensionality.
Yeah.
But I'm wondering whether this is not self-inflicted, because you decide to represent an image as a high-dimensional vector. You could as well decide to represent an image as a function of two variables, like expanding in a wavelet expansion, which is used for encoding and other purposes in image analysis. This would be a low-dimensional problem, and it would perfectly encode neighborhood relationships. So why wouldn't you base your training of the regression in a straightforward way based on those? You wouldn't get a log to the 6th power in the estimates. So, and at the same time, you are less constrained by regularity. For instance, you say these guys have to be Hölder continuous. They would like to have some Hölder continuity, which is kind of limiting the sharpness of contours. While already a very low degree of Bessel regularity would put you in a compact set where you can get a predictable convergence rate. So why are you going into this high dimensional?
I think this is very nice questions. So at the beginning, our motivation is to try to see that when X belongs to the R2 or R3, whether we can use the DNA method to try to develop the conversion rate. And then we found that the conversion rate we get here can be free of the dimension D. So that's our first motivation examples. We know that there are many other methods to try to dealing with the imaging data for the 2D or 3D. But in the context of the functional data, right now, as our best knowledge, there is no other available method also doing the same thing to use the DNN method. So try to solve the curse on dimensionality is one of the motivation. And the other motivation is we try to see if we can use the novel DNN method to play with the functional data analysis for the 2D and 3D imaging data. So the motivation data set is 2D and 3D, but the method can be worked for any dimension data set.
Okay, thank you.
Okay, yeah, thank you very much. Any other questions? If not, okay, thank you for the presentation. Now the next talk is Andrew. Andrew, please start sharing your slides.
Okay, can everyone hear me? Fine.
Yeah, sure.
Excellent.
Okay.
All right, cool.
Very
Let's go
nice.
ahead.
So Next talk is Andrew Hinton from Chapel Hill, so University of North Carolina there. So he will talk about perturbation in gut microbiome composition in response to food protein, predicts anaphylaxis in genetically susceptible mice. Okay, so very long.
Sorry about that. That was a mouthful.
Oh, that's fine.
Yeah, so today I just want to talk to you a little bit about a project we worked on where we were specifically interested in measuring the gut microbiome as it relates to development of food allergies. So as you mentioned, I'm a student here at UNC. I'm in the labs of both Peter Mucka and Wesley Burks. And I want to go ahead and just get started. So just kind of to get you sort of a good feel for what we're going to be talking about, I want to talk a little bit about food allergies. And so interestingly, the top food allergies or top food allergens are peanuts, tree nuts, milk, shellfish, eggs, soy, fish, and wheat. These are sort of notorious for causing a severe reaction, and about 90% of all food allergies are in general related to these allergens. Interestingly, these are common in kids. So 4 to 8% have been estimated to be affected by this. In fact, my daughter, which is part of my motivation for sort of studying food allergies, suffers from peanut allergies. And as you know, if you've probably seen on TV, they can be fatal in certain situations. And even more interestingly, it has recently been estimated that about 11% of all adults have some type of food allergy, and more importantly, 38% have been shown, or at least have noted through various surveys, that they've had at least one trip to the ER based on their food reaction. So what I'm hoping to kind of demonstrate here is just that the food allergies are really a problem. And then more recently, the field is shifting in a way where where there's some thought that the gut microbiome may regulate sort of the immune response. And so some of the immune training that goes on happens directly in the gut. The gut is loaded with immune cells. And so the intestinal sort of microbiota may be really important for sort of regulating this. So maybe there's something in the early gut that maybe helps protect or make one more susceptible to developing food allergy. And more importantly, and I'm not going to talk about this in this study in detail, but this idea that IgA, which is a particular antibody, is also really important. And in our paper, which I'm going to show you a little details from that, we focus more on that, but this talk here I'm not. So I'm also in the topic, or in the title, you sort of saw that I mentioned in genetically susceptible mice. And what I mean by that is that in order sort of to study the food allergy and the gut microbiome, we need a model. So like not a model in a sort of a computational sense, but more of a mechanistic model to study these things in mice. And so at UNC, there's this project called the Collaborative Cross, where a bunch of mice from genetically distinct backgrounds were sort of like inbred, and they inbred, and then eventually you get these really cool and interesting diseases. In particular, our lab recently be able or to have the same type of sensitization route. I am unstable. Okay, it looks like I was unstable there for a second. But the CC027 strain has been shown to sort of become sensitized in a similar manner to humans, in that orally they can be challenged and react. We don't see this in typical laboratory mouse strains, and so the CC027 has more of a human sort of tolerance and reaction sort of propensity, and given that it's really cool and interesting to study allergies in this model. And so in comparison, there's some key or critical immune differences between a strain that we're going to be studying, or sort of like comparing the CC027 to, in that the C3H strain of mice. They have fewer T regulatory cells and more intestinal mast cells. Mast cells are really important for developing food allergies, and so that's kind of what that's listed there. And so I'll just talk about the experimental design, because before we actually start doing our model and those kind of things, I'll just kind of ground you in how we did this experiment. And so in this work here, what we did was in the first 4 to 6 weeks, we took mice from both strains, so the CC027s and the C3Hs. Fecal pellets were collected before they were exposed to the food antigen. So then, and then they were collected after they were exposed to the food antigen. And so after this sort of first week, then we slowly introduced protein or food protein and in that peanuts, walnut, milk, egg, and so on. And then we collected that. So this is kind of where our data is going to sort of give rise or come from. And so again, just a little bit more background before we get into the model. So this was 16S sequencing that was done on the microbiome. On the fecal pellets, they were flash frozen and they counted them. In particular, we got count data from the Illumina MySeq platform. The OTUs and ASVs were, and that is amplicon sequencing variants, were assigned using the data2 pipeline. And then the taxonomic assignments were made using green genes. So when we started to label these things and identify what bugs or species or strains were there, they were done using sort of the phylogenetic matching to green genes. And then ELISAs was to use to quantify IgA. So in my work, I focus a lot on this particular problem of the microbiome data existing. In that is count data, it exists as compositional data. And I don't have enough time to go into all the details of compositional data, but what I would like for you to take away is that compositional data are different from our normal sort of like data in Euclidean space, where you can range from negative infinity to infinity along any axis within your dimensional, whatever dimensional space. Whereas in the simplex, we have certain limitations and restrictions. And because of that, we have to address this in some way. And so the count tables are made to be relative abundance. So in a microbiome study, you've probably seen or read, many folks will reference relative abundance, but those data are truly complicated. And so in particular, so we can map the count data onto the simplex using this closure operation here. And essentially this converts it to relative abundance. And then in our case too, I started with these pre-exposure and post-exposure matrices, which were in samples across 230 different taxa that we identified. And then after we close and sort of map it onto the simplex, we have to, because we're interested in doing a log ratio transformation, so we have to handle the zeros. We can't take a log of a zero, or you can't take a ratio with a zero in the denominator. So we use a multiplicative replacement strategy. Again, not enough time to go into detail of this, but you can basically think of this as a smarter way to impute zeros rather than adding a just random pseudo count. And then we finally get our pre and post exposure matrices. Excuse me. And then from those, what the title also mentioned was a perturbation. And so what we're really interested in, because you can't directly measure how the gut microbiome changes from a pre or post if you have count data, we can leverage this cool concept from John Atchison called the compositional perturbation operation, which is where we essentially take this closed data after imputation and we perform this operation, which is essentially dividing the two matrices and reclosing them. And that's what gives rise to our perturbation. And so what that means in this concept is we're actually measuring the way the microbiome changed in response to food allergen. So now we have this pre-exposure, we have this post-exposure, but now we know specifically when a mice was exposed to food, whatever, how did that microbiome change? And so what we're interested in here is, did that microbiome change in those that reacted? So of the mice that did react, did it change in a very specific way? Was there some madness or method to the madness in that the way that the microbiome changed? And so what I will note, because we did mention earlier that we have the C3H mice, and so what this slide ultimately is trying to demonstrate is that really only the CC027s reacted. And so for the rest of this analysis, we're only interested in the CC027. What I would also like to show is that the pre-exposure microbiomes were drastically different between those two strains. And so like using the C3H may not be helpful because one, we know they don't react, and two, we know the microbiomes were entirely different. So that's what this demonstrates using just some hierarchical clustering is unsupervised and everything. So no labels were examined. So just moving on quickly to the models. So we basically use partial least squares discriminant analysis, and we trained it using 20 repeats of tenfold cross-validation. This is a really small sample set. It was only 48 samples. So you imagine that these estimates can have a lot of variance. And what you can see in general is that we have some level of prediction using the pre-exposure microbiome. Interestingly, when we use a post-exposure microbiome, we're not as, the prediction ability goes down. But because I mentioned this perturbation operation, I'll close with this, is that the perturbation operation seems to improve the reaction in that the way that the gut microbiome responded in response to the food treatment or food protein was different. And so in general, what this talk is trying to at least convey is that there is some interaction between the immune system and gut microbiomes, and that can potentially influence things. And that specifically changes in the microbiome may be predictive of future allergic responses. This is the paper if you're interested in kind of reading more about what we did. This perturbation analysis here that I went over today is not in that paper, but this is something we've been working on. And the code, and if you want to pull these data down, you can find it here on my GitHub. And with that, I'll close. And this is my lab, the funding sources and everything like that. Thank you.
Okay, thank you very much. So any questions? Can I ask you what is the way you actually process the data? Do you use deep neural network or setups?
So I use partial least squares discriminant analysis work really well here. You can train deep neural networks, but because we have so few samples, we run into the problem of probably overfitting. Becomes really challenging in these data. And also too, if you select the right set of ratios, the space tends to separate linearly. And then you have more interpretability with the PLSDA, which is why we did that. For some other projects, we do try to leverage convolutional neural networks and those kind of things. But for this specific one, we went with partial least squares discriminant analysis.
Yes. So simply, I'm asking this question because you have compositional structure, and that's probably very well fit to the neural networks. That's why I was asking. So.
Okay. Yeah. Thanks.
Yeah. Other questions? Yep. Good. Thank you very much.
Thank you.
So now we're up to the last talk for this morning session. So this is Jeungeun Park from, can you start sharing?
Yeah, sure.
Yeah. Okay. So that is from University of Cincinnati. She will talk about corrective behavior in bacterial chemotaxis. Okay.
Yeah. So can I start right now?
If you want to, you can tell a joke if you want to.
Yeah, I think I can start. Yeah. So I'm Jeungeun from the University of Cincinnati, and thank you for giving me such a great opportunity to enjoy this spring school and to introduce my research work. Yeah. So I'll talk about collective behavior in bacterial chemotaxis, and this is a joint work with Zara Emenzer at the University of Iowa. So before I talk about the chemotaxis, let's see how an individual cell swims. So I'll give you an example, E. coli bacteria. So as you can see here, maybe I'll use some. So here, so E. coli has multiple flagella over the body, and each flagellum is connected to the cell body via flagellar motor. And if all of the motor rotates counterclockwise, then it forms a bundle, flagellar bundle, and they run. But if one of the motor change the direction, like if this turns clockwise, then they start to tumble. It means they change the direction. So if there is no chemical signal out there, they respond, then it usually takes 1 second for a run, and it takes 0.1 second for tumble. However, if we introduce some chemical signal they like food or nutrient, then they are less likely to tumble, or the mean length of run becomes longer. It means in the end they move toward the food they like. So such a movement in response to chemical signal is called chemotaxis. So as you can see, I've talked about only one single cell chemotaxis. However, in practice, collective behavior in chemotaxis is very important. For example, how the cancer cells spread helps to aid the diagnosis and treatment. What's more, when the cell aggregate, then they show interesting patterns like traveling wave, or sometimes they show exotic patterns. And here, light colors represent the high cell density. So likewise, I'm interested in collective behavior in bacterial chemotaxis, especially one of my research project to answer the question, when we have the data, which is described at the level of the individual cell, or microscopic data describing individual cells' behavior response to chemical signal, then how can you understand collective behavior? This is what I want to answer the question. In other words, I want to derive macroscopic model for chemotaxis from the microscopic model of the behavior of individual cell. Yeah. So to answer such a question, some may say, could it just simply track each single cell in a population or in a kind of group? But this is kind of computationally impractical. Another suggestion could be, could it just use well-known classical PD model, which called Keller-Segan model, to describe chemotactic behavior. But some parameter, this Keller-Segan model contains some parameter and sensitivity function which measure the strength of signal, chemical signal, but these are phenomenologically chosen. So it is difficult to guarantee the choice of the parameters or the shape, the function for sensitivity functions. So that's why many research groups suggest some mathematical framework to drive an equation of the cell density. I'll denote the cell density here by n from the microscopic model. Yeah. So to do this, I'll emphasize two important factors which describes the cell's cells movement in response to chemical stimuli. So one thing is velocity jump process. As you can see, if you recall the movement of E. coli with the same velocity they run, but instantaneously, randomly, they change the direction. So these can be modeled, often modeled by velocity jump process. And the other thing is these kind of rate of change, the rate of change of tumble or the tumbling rate depends on their internal or intracellular dynamics. What I mean by this is once the cell detects the signal via their signal receptor, then something is going on inside, but this is called a signaling pathway, but it looks very complicated. So main key here is to reduce this signaling pathway to variable or some function A, which called internal dynamics. And this internal dynamics is described by an ODE. So, and via this signaling pathway, this determines the rate, the tumbling rate, yeah, whether they change the direction of the motor or not. So if I use jump velocity and internal dynamics, then if I introduce the cell density, not only depends on the location and time, but also depends on the velocity they move and internal dynamics, then we can jump velocity jump process gives us some equation, which looks like as follows. And if we introduce macroscopic density of cell density of cells, and by integrating this P over all possible velocity, all possible internal dynamics, then we can finally define the macroscopic variable density of the cells. And then using this and by using some mathematical tools, then we can derive some advection diffusion equation as follows. Yeah, even though this looks like classical Keller-Segal model, but I want to emphasize that when we derive this equation based on from the microscopic model of the individual cell, then these parameters are describe the internal, depend on internal dynamic and depend on the gradient of the signal. Yeah, this is what I want to emphasize. Yeah. So basically, my work with Zier is inspired by the following paper. So this is experimental paper describing a collective behavior of the cell when we have two different signals on the left and right, then some move to the left, some choose the right foot. So based on the exactly same experimental setting and microscopic model for individual cell, then we have two simulations, one from Monte Carlo simulation, and the other one is PD solution that we derived earlier. Then as you can see, we have almost the similar, we have kind of good agreement each other. Furthermore, when we compared our result with experimental result, and we found that they are qualitatively agree very well, but we can improve further. One thing is this, because when you use a internal dynamic here, these days many researchers try to kind of improve this part. They want to answer how they combine information optimally from a spatial array of the signal receptors. And the other things, we can improve our model by considering some communications between the cells. Yeah. So this is about the kind of one of my ongoing research, and yeah, that's it. Yeah, this is all for my presentation for today. Thank you.
Thank you. I think Wolfgang is eager to ask a question.
Thank you very much.
Thank you.
Interesting, but maybe at some point I lost you. I didn't quite understand. So the model you described, which was that transport equation coupled with the global operator that represents the probability of tumbling.
Yeah, right. Yeah.
So it wouldn't be, but in the end, you say you reduce it back to this classical equation, which is only a PDE, is it? Or what am I missing here? So that global part is no longer there. I thought originally you would use your data to learn that kernel in that global part. Is that? But that's not what you do, right?
Oh yeah, yes. Actually, here to drive this equation, we use the simple assumption on this T. Yes, as just constant.
Or you basically you simplify that. You
simplified, yes.
To an average, right?
Yeah, right.
I see. But that may have a gross effect on your model, don't you think so?
Yeah, right. Yeah, right.
I see. So would it make sense from your point of view to use data to train that kernel with a little more accuracy than just reducing it to an average?
Oh yeah, that's true. Yeah.
Isotropic, and then it doesn't really distinguish between directions, right? So yeah, I wonder how you get this streamlined formation when there is an interesting signal out of this kind of global term. But I may just misunderstanding it. Sorry.
Actually, I dropped all of kind of details here. From here to the differential, the PD part. So
okay, no, don't never mind.
Well, yeah, if I have time, I'm gonna show you how I drive. Yes.
Thank you.
Yeah, thank you.
So, but I just want to say in general that it seems very interesting problem in which you can learn the behavior and actually create a model based on data. That is probably a test bed for some kind of this type of research, which now it's very modern because people say, okay, we don't want only to learn the solution of differential equation, we want to learn the differential equation itself.
Yeah, right.
So that's why probably that's the interesting direction.
Yeah, I think so, yes.
Anyway,
thank you.
I want to thank all the participants in this short session. So everything was fantastic. So thank you very much. And don't forget that there is a talk at 2 o'clock, and also Wolfgang wants to say something.
Yeah, Peter, don't shut off immediately because Mirjerta wants to discuss a question.
Oh, okay, okay. Should we continue recording, or Mirjerta, do you want to record, or that's just stop now because that is the official session? Or do you want to record?
It is totally fine. It doesn't have to be recorded. It's okay.
Okay, I'll stop recording then. Okay.